Thank you very much. I think we'll just get going and people can slide in through the doors that are still open as we go. Thank you very much for joining us. Really excited to present to you some of the work that we've been doing.
I'll start with
our
experience on the utility of the toolkit for correction.
We'll start off with me figuring out how to use the remote, which is not going particularly well, I have to say. Thank you. So all meetings are being live streamed. There are the virtual platform who are watching, and there will be recordings that will be later made available. They will come online a week today. Live streams will pause whilst we're doing the breakout sessions, which is good. And the smaller group discussions, we will be working round the room with some questions for people to give their insight in certain topics. Breakout sessions, there is no interpretation feature, so bear with. And as we have done for the last 2 days already, please take a moment to observe where your emergency exits are. So we've got a few presentations today. We are focusing on one project that we are operating together. We have talks coming up from the people who have been implementing the research in the field. Overall, our session aim is to present to you what we've done, how we've gone about doing it, and then get your insights into what could be done better, how we can improve, what we've missed, and of course, further research questions that could spin off out of it. Thank you very much. Great. So I'm going to hand over to Diana from Glide to say a few words. Yeah. So over to you.
Thanks, Richard, and thanks everyone for joining our session today, and thanks to the people joining online as well. So just to introduce myself, my name is Diana Youssef. I'm a technical advisor at Glide, the Global Institute for Disease Elimination. For those of you who haven't heard about Glide before, we're a global institute, as our name suggests, focusing on disease elimination, and our priority of diseases of focus at the moment are onco, LF, malaria, and polio as well. We're a young institute based in Abu Dhabi, and we were founded in 2019 through a collaboration between His Highness Sheikh Mohammed bin Zayed, President of the UAE, and the Bill and Melinda Gates Foundation. And so our mission revolves around 3 main goals: elevating awareness and engagement in disease elimination, advancing disease elimination strategies, and fostering and scaling innovation to fill gaps. And I feel that this DISSECT work that you're about to hear more about today really aligns with our goals, and it's one of our bigger pieces of work at Glide. We are currently the sole funders of this work, and we work in partnership with Sightsavers and also country partners as well. And so that's a little bit from me, but speaking of country partners, I'd like our country representatives to please introduce themselves as well. Thank you, and with that I'll hand over to Richard. We have a few presentations, and so Richard will give introductions to our next speakers, and then after the presentations we'll break out into the group discussions. Thanks.
Thank you. So all of this work has stemmed from the WHO entomological manual. I'd now like to invite Maria, the lead for Onchocerciasis and scabies, to come up and talk us through the entomological manual. Thank you.
I'm not sure if these lights can be switched off or no. I don't know, just because there's quite a lot of light in the thing. Excellent. Thank you so much for being with us this afternoon. let me know if I speak too fast, because I always have that problem. So I will try. So as you know, this year the WHO released a new manual for entomology that was a long due project. I think we all, for the many, many years, have been really looking forward to have something that could serve to train countries on how to improve the entomological surveillance, the surveys, and the use of entomology in the field for onchocerciasis elimination. So in this brief overview, I'm just going to outline the chapters of this manual so that you can go to online and download it and really study all the details of the manual. But just to start, I want to start by framing, this is the graph that you all know very well, the cycle of onchocerciasis elimination. And I want us to reflect for a second what flies tell us and what they cannot tell us. What is the value of entomology and how we can use it together to make decisions at program level. So if we see this graph, we can see how our baseline prevalence, this is the prevalence of adult worms in a certain population, and how there is a decline as we start with our interventions, there is a decline in the adult worm population until we reach a moment where all the adult worms are dead, and it is safe to be able to stop interventions, because when we stop, there is not going to be any resurgence because there is no adult worms remaining. So before we reach that point where all the adult worms are dead, there is a moment where transmission is suppressed. And transmission is suppressed when all our flies become negative and they are no longer transmitting the disease. That happens many years before we can actually stop NDA. So our flies become negative many years before we can actually safely stop NDA. And I actually like to highlight that because often in the field we interpret having negative flies at it is the safe time to stop NDA because there is nothing in the fly, so we can stop NDA. And actually that is not true, and we all know that, no? If we stop NDA when transmission is suppressed and our flies are negative, we will quickly have a resurgence of the disease because our adult worms will still be alive and they will restart the production of MF, microphiladermia, and we will see how they will become positive in some years. So that tells us that we can use entomological surveys to assess when transmission has been suppressed or to really use it as a surveillance tool later when we stop NDA to see if there is any resurgence of the disease transmission. Those are the two utilities of our entomological surveys. And it's something that needs to be a little bit improved in the countries because often we are not using it enough for monitoring and evaluation and understanding when we reach the point of transmission suppression, and we don't often use it enough to assess post-treatment surveillance, to really do the epidemiological surveillance, the surveillance in the field to see if our flies continue to be negative after our interventions have stopped. So those are the two moments where entomological surveys are critical. So with that objective in mind, we have published a training manual for health workers or for programs to use to improve. So oops, it's going fast. It's going faster than me. Where is the? Good, excellent. So this is the reason why WHO decided to publish this training manual. Because, as we said, it has a very clear programmatic purpose, to be able to use this tool to assess transmission suppression or to assess any resurgence of the disease after treatment. Unfortunately, we clearly all know that there is lack of skills in the field of entomological capacity. There is very few entomologists, and it was important to be able to develop a training manual that could be utilized at country level. The manual will represent a tool for strengthening capacity of scientists and in all the areas of evaluations and surveillance. And now, what was the methodology that was used to develop the manual?
So what's in it?
Of Nigerian colleagues
the manual is
started
174 pages long
then it was further,
and is available in English.
Revised by several,
it is currently being translated into French,
the document is
Portuguese.
174 pages long. And we had expert review panel from 10 countries reviewing the document to try to improve the different chapters, the different elements from 4 different continents. And a critical review of the first draft then resulted in further edits, further changes, and a development of the manual. And the entire funding was provided by the Mectisan donation program, and we are truly thankful for that.
So what's in it?
We have 12 chapters, and I will
The
explain
manual
what is
is
the content of each of the
174
chapters.
pages
I did
long
put the reference
and is
here
available
how you can
in
download
English.
it. It's not printed
It is
yet.
currently
We hope
being
to print it in the
translated
future,
into
but for now,
French,
it is really
Portuguese,
downloadable, and
and
you have to print it
Spanish.
in your house.
There are
It's gonna
12
consume all
chapters,
your ink, but,
which I'll briefly review. 122 references.
Let's see. Oh, voila. So the first chapter is really the background and the learning goals. We have, why this thing goes alone? I don't understand. Maybe there is some setup situation or something, I don't know. So we just start with the usual background, abbreviations, acronyms, some graphs, and because it's a training manual, each chapter is preceded by a learning outcome. So it's really not the traditional guideline, it's something more usable for learning. No? So it has the learning outcome and how to assess and evaluate the outcomes. The first one is on vector biology. So we try to describe, okay, you can continue doing your own thing. There is a description on all the different types of simulion, the blackfly vectors, what are the types that apply in each of the geographical regions in the world, so what types of different blackflies we have, how to identify the blackflies that are vectors versus the ones that are not vectors. Then we enter to how to prospect the breeding sites. What is the methodology to identify breeding sites, what we call breeding sites assessments. How to mobilize the community to conduct those surveys, how to preserve the blackflies so that you can take them to the lab for conducting the cytotaxonomy. The next chapters, and we are already in the 8th to 10th chapter, it's driving me crazy, is the methodologies for the capture of the actual flies. Once you have actually identified your breeding sites, you, depending on what is the purpose of your collection, you will select the community where you think more infected individuals will be, and that will be the place to do the collection of flies. And there will be a description of the different methodologies that can be utilized, from human landing collection to the different traps, to and how then to go to the dissection of the female blackflies to determine parity and infectivity, to prepare these blackflies for the PCR, and then to ultimately determine whether they are infective or not. And we are coming to the end of the manual, where we have chapter 11 and 12, where it helps you to determine and interpret the transmission indexes. So the results of your PCR will help you to identify what is the transmission index for onco-elimination and whether you have reached already the threshold where you can consider elimination or suppression. Again, transmission suppression has been reached and there is actually no active transmission. And really, this was just a very quick overview because it was not supposed to be a training, but I really encourage you to download the manual, to read it, and to extend it to all your colleagues and to make it sure that all the countries have knowledge of this manual. And also send us the feedback if you think different tools could be complementary to this manual, what is missing, what can be done to use it better. Thank you so much.
Thank you very much. So I'm Richard Selby. I'm one of the co-PIs on the DISSECT project, which I'll give an overview of now. The other co-PI is Louise Hamill, who is joining us online. She's the technical director for Onchocerciasis and lymphatic filariasis at Sightsavers. So the DISSECT project has a meaning, developing innovative scalable solutions to entomological gaps and cross-border transmission of onchocerciasis. The rationale for it coming about has been there is a lack of operational guidance for standardizing entomological data collection. I think most people in this room will agree there's a need to replace HLCs with something that is, number one, safer for the people doing it, and number two, more standardized between individuals operating. We're relying on outdated methods of breeding site identification and going about monitoring them as a result. We need to improve the capacity for analysing entomological samples that have been collected within the country that they're collected, or at the limit within the region they're collected in. A significant minority of Onchocerciasis foci require alternative additional strategies to aid ivermectin MDA. And finally on this list is there are transmission zones that span international borders, which presents a whole raft of additional challenges. Working towards closing some of the gaps around this, if not all, is the DISSECT project. So we're conducting research in 2 study areas. They are cross-border areas, so in total we have 4 countries. We have Malawi, Mozambique project area, and we also have a Côte d'Ivoire and Ghana project area. GLIDE are not only funding this work, they're also partners in all of the research. The Ministries of Health in all 4 countries are also research partners. They're heavily involved. The research is focused on solutions to the challenges that the programs themselves face when monitoring Onchocerciasis, skewed towards the cross-border areas. That's where we're starting out. The entire project has a duration of 28 months. We started in January last year, so we're getting quite far along. And the research activities that we're doing is generating data in all 4 of the countries, so both study areas concurrently. All of the research aligns with and strengthens the planned routine Onchocerciasis monitoring and evaluation in all 4 nations. Here's a map of our study areas. These are our actual ending study areas refined from this. This is the initial start point that we went to the national partners with and kind of refined it from here. So you can see we're kind of in the center of the border of Côte d'Ivoire and Ghana, and Mozambique, Malawi, we're in southern Malawi into kind of the eastern side of Mozambique. Our project's outputs, we have 4 principal outputs and 2 kind of secondary outputs. First of all, we're working towards a standardized entomological data collection toolkit for breeding site identification and transmission assessment. This portion of the work has been completed with Mozambique about to finish the transmission assessment in full. We now have Mozambique and Malawi actively doing the optimized blackfly trap development. I'll talk about that in a little bit. And following on from that is the third point is trying to work towards an updated approach to breeding site prediction and validation. The fourth one is developing regional centers of excellence for programmatic analysis of entomological samples. Underpinning these principal objectives, we have 2 additional secondary ones. First of all is once we do have a trap developed, a viable trap developed, understanding the potential for that trap to ultimately be used as vector control. It's wishful thinking maybe, but we'll never get anywhere if we don't try. And secondarily, understand the opportunities and challenges with integration of this blackfly trap with other insects and diseases. I'd like to highlight the technical steering group that we've got. So every country, so all 4 countries nominated their own member of, their own representative, to be a member of our technical steering group. They've had access to everything that we've done. They've been sat on our meetings. So on top of that, the Ministry of Health employees in every country, teams in every country, have worked incredibly closely, not just with the national committee member, but also with the research coordinators in each country, many of whom are in this room today. So our approaches for standardizing the entomological data collection toolkit, we can summarize it into, we took the WHO entomological manual and we have developed a toolkit for refining it for the study areas and then ultimately the specific nations, and I'll give a quick overview of that process. The toolkit has been trialed for 3 activities: breeding site assessment, collecting of dry blood spots for serology, and validation of adult fly populations, both by human landing capture and Esperanza window trap, following all of the guidelines that are in the entomological manual. So, we were very kindly provided with a late draft copy of the entomological manual, which came out in July this year. as Maria has already highlighted, it is 174 pages in length. So what we did was we took that document and maintaining the structure of it, we iteratively, myself and Louise and other team members started by removing things that were relating to Yemen and South America and everything else that we weren't going to find in our project areas. So we refined it down to locationally relevant content. We then took that to, first of all, a cross-border meeting, and we presented it to representatives from both countries, got their input, circled back round, implemented all of their changes, and ended up with a finalized document complete with example forms for data collection, which was running at around 45 pages, depending on whether we had 1.5 or 2 line spacing. So that was then the document that was the toolkit, sorry, that was refined for the study area. We then had to get a national refinement, and this is an image that Rogers has provided from Côte d'Ivoire. So the standardized toolkit for Côte d'Ivoire and Ghana was given to national experts in Côte d'Ivoire who suggested some more changes that were needed to get it through the ethics committee to have our ethical approval for DBS for HLC. We then took that, got our ethical approval, got our draft toolkit approved. The text is a little bit difficult to read here, but we then took it to implementation. So we then went forward to go into the field, finding the breeding sites that we wanted to go to, doing the breeding site assessment, collecting dried blood spots, collecting collecting adult flies. After that first phase, we've held pause and reflects in all 4 countries. We've got the opinions and experiences collected straight back from the people who are involved with it and other stakeholders, experts in the country. And that has then been fed back into our secondary ethical approval for the refinement of traps. Speaking of refinement of traps, so for the trapping element, we are going through a series of optimizations to end up ultimately at 2 or maybe 3 optimized trap prototypes for each study area. I'll do a quick kind of skipping stone over some of the things that we're doing in a bit. All of the trap development that we're going to do will start out by this kind of rotation of different trap variants to see which ones are most attractive to the blackfly. We are going to do everything with a HLC in as part of the random rotation, so we will be able to compare the trap efficiency to a HLC. Once we have our optimized traps and we know what is most attractive to the blackfly, we will then trial using them at scale to see essentially how many traps we need to put out to get the number of flies we need to do the assessment of transmission. So here are some of the trap iterations that we're going through. We are varying the shape of the traps. We are varying the color of the traps, so we've got all blue, all black, and then blue, essentially very similar to an Esperanza window trap and an Esperanza window trap plus a UV light. And then we've got 2 different variations of scent. So we've got the same 4 scents in both of these. So we are doing a BG lure, socks, warm socks, obviously not clean ones. And then we've got 2 chemical lures that we have selected, one from a Cozart paper and one from a Young et al. Paper. Now, we've got 2 sets of these because one of them we are baiting with carbon dioxide being made by yeast, sugar, water, which we know works, but it is incredibly difficult to do at scale. I think we bought 300 and something kilos of sugar in each of the nations, which must have made some people at the market very happy. The second load, we are using carbon dioxide lures, which have been supplied to us by Chemta. So thank you very much for the collaboration there. And so let me get this right. So one of them is the Young et al. The bait sat on what are they called? Odor absorbers, and the other one is scent beads. Thank you. And the other one is the Cozart one, which is in plastic bags and then punctured. Updates to breeding site prediction. We are using the evidence that we are gathering to go into the remote sensing. We are aware that we're not operating on this alone. It's good that we are collaborating with other actors in this sector, and we would like to kind of get more input and engagement on this. So I know there's going to be a lot of interest, so that's welcome. So we're looking to push forward geospatial analysis and modeling on what we've done and where we've been. I'll show a couple of images on that in a minute, and the rest of the team will dig into it with their country perspectives soon. So we'll then look to have them run through an algorithm to outcome with a breeding site locator tool to refine and give feedback. The outside, on the back end of this, we are going to go back to the field, and with this, the projections of where it should be good or should be not so good, we will trial it in the field and validate this step. So the BSA that we've done so far, in total, oh, sorry, I should expand on this. We have the DISSECT areas here and here. But in addition to our DISSECT area, reacting to the fact that Côte d'Ivoire had not had a national BSA for a considerable amount of time, Glide funded the national program in Côte d'Ivoire through DISSECT to conduct a national level BSA. So combining all of the data that we've got very, very close to 750, 744 sites were targeted for inspection. We managed to get to 706 of those. Of the 706, 41% had larvae and 59% did not have larvae. So that's the data set that we are looking to train the model, and then we'll validate it later on separate field visits to where we think we can find or not data. The fourth principal objective is to develop centers of excellence. We know that there are talented and very dedicated scientists and laboratories in the countries that we're working in. There are challenges with getting supplies in for reagents, and also some additional training was needed. So what we're pushing towards is trained laboratories that have been validated with the staff equipped to perform blackfly analyses to support not only their nation, but we're fostering cross-border collaboration, and it's going quite well so far. So if there is a problem in one nation, they are friendly, and they can ask the cross-border nation for assistance. So that's what we're working towards. We have already carried out the lab capacity assessments. We've done some training on the adult blackfly and larvae and dry blood spots in particular, and we are on course with mosquito work as well. Our secondary objectives, that's not particularly easy to see on the screen, I do apologize. So investigating traps for vector control. So once we have our 2 or 3 most promising trap candidates, we're going to deploy them at scale in different densities around the breeding site and the kind of area that goes up towards the habitats that people are living in. We will leave them in situ for a prolonged amount, for an amount of time, and that will be bookended by HLCs, so we'll know before what the biting rate was and after what the biting rate was. We can model and analyze the results, and after that we can make recommendations for further studies that could be done to analyze the use of traps for actual vector control. This is a proof of principle that hopefully if it works we can build something out of it and maybe have some traps as vector control. Thank you. The last section that I'm going to go over is the potential integration with other traps and diseases. So we've collaborated with the Liverpool School of Tropical Medicine to do a situational analysis of other vector-borne diseases within both of our project areas. And we have, out of that, we've got the kind of culprit list of what we're expecting to find and the traps that we'll need to best find them. On top of those additional traps, we're also looking at the analysis of the bycatch from the prototyping and the finalized traps. So all of the data collection from that is not just picking the blackflies off, we're also picking off anything else that comes along, be it a midge, be it a mosquito, be it a tsetse fly. We are taking the sample, recording all the information. We're looking at the co-deployment, as I've already said. We've had the recommendations for the other traps. We're going to look at the co-deployment of those alongside blackfly traps. And by alongside, I don't mean next to, I mean in that community. So we have the ethical approval for doing combined trapping in a community. And we will finally be examining the operational efficiencies that are gained, as well as the complexities that are added by adding additional traps and taking off additional insect species and, genuses and trying to figure them out in real time. Thank you very much. I will now hand over to Mozambique and Malawi. Nelvina Makatu is representing Mozambique first. She's the research coordinator for us in the Mozambique project area. Unfortunately, John Chipeta from Malawi couldn't join us. He couldn't get here, but he's recorded a slideshow, and we'll go on to that afterwards. Thank you.
Good afternoon once again. So first of all, I'm a Portuguese speaker, so pardon for my accent. I'll try to improve it. So, in Mozambique, as Richard was saying, we were working with Malawi in a cross-border area. I think it's okay. In a cross-border area that he mentioned already. Basically, we're working in two districts that were crossing through Malawi border, that is Mulumbu and Milange. So we did two BSAs, and one was in 2022, yeah. Sorry. And we were just focusing on perennial rivers, and then we did this year a second BSA in both perennial and seasonal rivers. We divided our team in 4 groups, and we end up training a total of 16 people. Which team was having 8 people, like 8 entomologists and also 8 for HLC. As I said, we're working in 2 most productive sites, 2 in Milange and 2 in Mulumbu. Oh, sorry. And also we did trapping for 4 days in the phase. In terms of results, we had 163 sites targeted, and also we end up inspecting 19 rivers in 2022. From those numbers, we had a few positives and also negatives, but that was really nice for us to figure out how our site was, because Mozambique was just having data in Onco since 2007 in Remo. Then we had IOM in 2019. So in terms of entomology, this is the first data what we are gathering together. So a total of 3,911 flies were got in the site. And from these numbers, we had more fly bites during the afternoon. As I was saying, we collected 776 using HLC, and also we had 577 during the evening. The higher catch report was in Milange district, that is more close to Malawi. And so because of the weather, Mulumbu is close to the mountains, and the weather is quite different and rains a lot. So in terms of our experience doing the toolkit, the BSA, we end up understanding that definitely the HLC will be getting more flies. They are more attractive. We are humans. And flies, they're also intelligent. But we end up seeing that our HLC team will be exposed during that practice, and they can get also diseases. We gave them medication to avoid it, but we never know what will happen. In terms of the trapping, we got several species, and that is good with the traps, and also doesn't require a lot of experience because we're able also to train the community to handle it because they were so intrigued about the work that we are doing and they wanted to participate. The negative experience with the trap was, at least for us, was quite difficult to remove the fly from the trap because of the glue. And when we did the taxonomy test, we saw flies a little bit, sorry. So the general feedback around the toolkit for us is pretty good because we had a valuable insight from the toolkit in how to do the work in the field. We just needed to adapt it to our reality, and we did that with the Malawi team according to our country realities, procurement, and all the things. And also the toolkit gives the opportunity to bring two countries through NTD program together, fighting for the same problem, that is onco around that area, and also vector control and more understandable insights about the fly. With the toolkit also, we're able to understand more about onco and entomology data. In Mozambique, we are quite young doing that with blackfly. Unfortunately, we had Freddy cyclone in our study area that ended up changing the nature of the rivers, and everything. And that also messed up with the oncology of the vectors. And we are seeing some results in the second phase that we are doing right now. Okay, I think it's all for me. Thank you.
Thank you. It's not coffee break just yet, calm down. We're now going to cross over to John, who's unable to be with us, but he's provided a video where he's going to go through his slides. Could you press play, please? Thank you.
My name is John Chipeta, DISSECT Research Coordinator for Malawi. I'll make a presentation on the Malawi experience on implementation of the standardized practical toolkit. So I'll start with our experience on the utility of the toolkit for collection of breeding site assessment data. In Malawi, we have the rainy season from mid-November to early March. So we conducted the breeding site assessment in November 2022 for perennial rivers, because by this time, most seasonal rivers are dry. We had then to conduct the BSA assessment again in May and June for both perennial rivers and seasonal rivers. 3 teams of entomologists from Ministries of Health were trained to support the entomological investigations in 3 selected districts of Cholo, Palombe, and Molanje. So a total of 48 sites were targeted for BSA, out of which 43 were reachable for larva prospection. Out of the 43 sites inspected, 20 were from perennial rivers, and these were prospected in 2022. 23 of these were sites from both perennial and seasonal rivers prospected in 2023. Simulium larvae were found in 41 of the sites. with 54% of the sites reporting larvae abundance of more than 50. Our experience using toolkit for collection of adult blackflies. A total of 12 people were trained, 6 each to conduct human landing catch, and 6 to conduct collection of flies using the Esperanza window trap. We conducted human land catch activities in the 3 most productive sites from BSA for a total of 14 days. EWTs were also used to collect adult blackflies for 14 days. What were the preliminary results? Esperanza window trap results. We collected 3,234 flies using the 8 traps. About 16.5% of these flies were collected in the morning versus about 84% of flies which were collected late in the afternoon. Trapping was highest in Choro district compared to the other 2 districts. As for human landing catch, we collected 3,376 flies, just slightly higher than flies collected through the trap. Very slightly high, just slightly higher. Choro district reported the highest catch again using the human landing catch compared to the 2 other districts. Blackfly catching was highest in the late in the afternoon across all the districts. Our experience on collection of that blackflies through human landing capture approach, positive experience for human landing capture. Fly collection efficiency was improved when using putters and aspirators compared to catching tubes. More flies were captured late in the afternoon than other times of the day. What were the negative experiences? Fly collectors reported fatigue as they tried to maintain their catching positions. And I would also like to share with you our experiences with collection of adult blackflies using traps. We had a positive experience whereby we learned that early engagement with community and allowing communities to select fly collectors led to acceptance and ownership of the exercise. So we left the power to select volunteers to help us with collection of blackflies. We left that powers with the communities themselves. So they facilitated selection of volunteers to work with us. And the result was a very good ownership and acceptance of the exercise. What were the negative experiences? Traps had to be removed on a daily basis to avoid vandalism, especially in Choro district, which was a strenuous exercise. This was not the case with the other 2 districts where there was any potential of theft or some vandalism. Flies were drying while on traps, which made them difficult to remove. There is a need to think about a new tactic to collect the flies before they dry. General feedback on the toolkit. The toolkit is simplified into easy to follow steps, which facilitated the different entomological activities. Safety measures listed in the toolkit were followed and no injuries were experienced. We also recommend including additional safety items to the list in the toolkit. For example, We recommend adding raincoats because sometimes you do the work while it's raining. A carrier box. When doing this exercise, we carry with us a number of items. So we need something to use to carry all those pieces of items. Also important is the torchlight. We stop collection of flies around 6 p.m. when it becomes a little bit dark and it becomes difficult to see the flies on the trap. So the torchlights will be used to facilitate the process of seeing the flies on the trap and collecting them as they put them into the preservation bottles. Ordinary gumboots were not very convenient to use, especially during a PSA in difficult terrain like ours. We would recommend use of waders when prospecting rivers with deep waters. And also would recommend light sports shoes to use when when prospecting in rivers which have very, very long distances. So it becomes difficult to walk long distances using gumboots. We also recommend consideration to use drones for breeding site prospection where possible to reach extremely dangerous spots. Some spots along the rivers are dangerous due to various reasons like dangerous terrain or dangerous wild animals like snakes. This is the study area map. You can see this part is Malawi, and this part is Mozambique. So this is the project area. The red dots are representing numbers of larva abundance, while the gray and black dots are representing the number of flies captured through human land catch and Esperanza window traps. The bigger the dots, the higher the numbers of either larvae or flies. Thank you very much for your attention.
Thanks, John, and thanks to all our speakers. I think we're about at time for a coffee break, and so far we've heard from Maria, an overview of the WHO Entomology Manual, an overview from Richard on the DISSECT project, and the Malawi and Mozambique experience on the implementation of the DISSECT standardized data collection toolkit. So after the coffee break, we'll have one more presentation to hear about the Ghana Cote d'Ivoire experience and lessons learned, and then we'll break out into our group discussions. So thanks everyone. I think we're just at about time now, and I know that the closing session is about to start. But thank you all for participating. I think we were going to have a report back, but I know a lot of people still haven't had the chance to look at the DISSECT toolkit. So please do have a look at that and get back to us with any comments that you may have. Comments are welcome. And thank you all so very much. Yeah, and we hope that you enjoy the rest of your day. Thank you.